All right, today we're going to look at some notes on basically we're starting population genetics and look at some of the things on how genes stay the same, how they change, and how we get to evolution. And this is all in terms of humans. So while frequency staying the same or constant seems boring, it actually serves some purposes in today's world. So, what is a population? It's an interbreeding group of the same species in a geographical area, um, like the West Siders is a population. Gene pool, collective alleles in the members of the population. So, all of the alleles that we bring in is our gene pool. Population genetics is the study of the genetics of a population and how alleles vary over time. And you may have heard the term gene flow. That's just basically migration and the alleles brought in through that. All right. We're not going to calculate any allele frequencies, so I'm going to skip that. But phenotype frequencies can be different in different populations. If we're looking at various disorders like PKU, so this is the inability to break down the amino acid phenylalanine. And if we look at the groups here, we can see that among the Chinese, 1 out of 16,000, Swedes, 1 out of 30,000, so fairly rare. Japanese, almost non-existent, 1 out of every 119,000, pretty rare gene. Among the Irish, Turks, and United States, gets a little higher. So that's those genes are in those populations more frequently. All right, microevolution. So this is how alleles change, which is not the point of this chapter, but it is the point of the next chapter. So allele frequencies change when we have non-random mating, when you're actually selective about who you have children with. Gene flow, so again, migration. If migration occurs, allele frequencies change. Genetic drift, these are basically populations go down in number, increase in number, go down in number. There's different reasons that this can occur. We'll talk more about that next chapter. Mutations, this actually is probably the least common one that occurs, but it does happen. And natural selection is unequal reproductive success, so you don't reproduce as much, so your genes aren't on. And if we get enough of these microevolution events occurring, we can then get a formation of new species, which is regular macroevolution. All right, I'm not going to go into too much on Hardy-Weinberg today. We are going to do a worksheet on that soon, but Hardy-Weinberg is basically developed by mathematicians. It's the idea where allele frequencies remain constant. Basically, all those things I mentioned before, the opposite has to happen. There has to be random mating. There has to be no migration, no mutations, no genetic drift, and no natural selection for these to occur. So this is just basically how algebra explains how the genotype and phenotype frequencies occur in equilibrium. These mathematicians, Hardy and Weinberg, had no idea about genetics. It was geneticists that looked at it and said, can you think this, and asked them if they thought it would apply, and that you get two genes from one for mom, one for dad that work. And we'll go through all these, the formula later. And so I'm going to skip through here, and we'll talk more about that next time. Why do we need it, though? Basically to determine carriers. That's the key thing, because we always know how many people actually have a genetic disorder. What we want to know is how many people are carriers out there. And we'll do that tomorrow. I do want to just point out with X-linked traits, keep in mind it's a lot easier for males because we only have one X. Therefore, we only have two variables to worry about. We don't have to worry about all of these up here that you do with females. But once you figured out the male, look, we got the P and we got the Q. So it's easy once we have X-linked traits are actually a lot easier. All right, why do we need to know these? Well, Hardy-Weinberg is very important in determining DNA profiling. So basically, while our genes that express phenotypes are not a big deal. Those things change. We can't do Hardy-Weinberg on them because humans are not randomly mating. They're not. They are migrating. Genetic drift has occurred in our populations a lot. Natural selection occurs. So what it does affect is things like introns or the junk DNA of our chromosomes, which remember, if that's 98.5% of your genome is junk DNA. Hardy-Weinberg does apply there because nobody's going around saying, hey, you got some cute junk DNA. I want to go out with you. No one cares about that stuff. So it is subjected to Hardy-Weinberg equilibrium. We can actually calculate the probability of having these genetic combinations in your junk DNA, and that's how we do DNA profiling. So we're looking at these small repeated samples of your DNA. That's usually what we're looking at. So when we look at individuals, we can see how many copies of certain genes they have, and depending upon how they look on gel electrophoresis, like here we have 2 bands, here we just have 1 because they're both the same, and here we have 2 bands. But these are this one's bigger, so it's going to be running slower in a gel electrophoresis. So we would see these differences in these DNA repeats. So DNA profiling was developed in the 1980s to identify individuals, and we use it for lots of reasons. Forensics, for either criminal investigations or who's the victim, things like that. During 9/11, just finding bits of people, that was all that we could use forensics to figure out who was who. Agriculture, there's a lot of genetically modified organisms out there. DNA profiling is used to make sure you're not stealing somebody's agricultural property. Paternity testing, are you my daddy? That's a big deal there. And historical investigations, I think we talked about the Jefferson family, among others, in Chapter 1. And keep in mind, DNA can be obtained from many, many sources. They're around all over the place. So all you would do is take this DNA, run it on a gel. The longer the piece of DNA, the further up the gel it is, and it creates different banding depending upon what you have. Generally, this is what it would look like. So we would have one person who would be the victim, and then we would try to match it up to DNA that we find from other sites. So this is basically what they would be looking at. Nowadays, they would run the gel and then have a computer read it, and it would figure out the read. So let's just look at a little criminal case on how a sneeze identified an art thief. So basically, this thief stole a painting. As she was removing the painting from the wall, she sneezed. She sneezed on the wall. So they actually got her DNA from her sneeze juice. And they were able, first off, found immediately that this person did not have a Y chromosome, so they knew it was female. So they were able to figure out, hey, there was only 8 women in this area. So they were able to get DNA samples from those 8 women and compare it to the sneeze droplets on the wall. And if we look at any one repeat, like this ACT repeat seen on chromosome 4, 1 out of 60 people could have had that. Doesn't really narrow down much. This GC on 17, 1 out of 24. This AAG-CTA, a little bit more, 1 out of 1,200. But anyway, we have what's the chance of having that particular repeat on those chromosomes? Is this chance. But to figure out the entire thing, we got to multiply that together. So when we do that for these 6 repeats, it turns out it's, 235 trillion things. So really, there's nobody on Earth that have had this DNA sample. All right, how do we do DNA profiling? Often we use blood samples because it shouldn't be contaminated with anything else. Nobody else's DNA should be flowing through your veins. Keep in mind, only the white blood cells have DNA, not the red blood cells. We would cut up the DNA with restriction enzymes and then use gel electrophoresis fragments to line them up. And then those gels don't last very long, so we move the DNA to a nylon sheet that does last longer. They don't usually use radioactive probes anymore. They do have fluorescent probes that work a little nicer, and they're not exposing people to radiation anymore. But these probes would bind to the DNA. Radioactive probes were nice because you could just throw it up against some X-ray film, and the exposure would develop the film, and we could see the DNA file, and then it's permanent. Then we can use it to identify individuals. And then we just go and compare the sequences. So you just see how the gel line up. So DNA profiles need databases. So we need to collect a lot of DNA information to see how many of these repeats are out there. We can use not only nuclear DNA, but mitochondrial DNA. We don't need a lot of DNA. If we don't have enough, we can make more through the PCR technique, which I think we went over earlier in the quarter. And we can do this on a large scale analysis. And those are just some of the examples there. There is some concerns about genetic privacy. Are we going to do DNA dragnets like they did in Gattaca, where they just stop people and collect their DNA right away? They do say that anybody who walked in a police station in England is automatically given a DNA test. Why they're doing that, I don't know, but they apparently don't have the same privacy laws that we have in the United States, because we do have HIPAA. When you guys hit 18 and start signing your medical forms, you'll have to sign this HIPAA Act, which means they can't give you information unless you request it. So there are some healthcare concerns because insurance companies would love to know if you have certain genetic disorders so they can charge you more. So that's another reason why we need to have privacy. And that basically ends this chapter. We'll talk more about the Hardy-Weinberg problems, how to figure it out. Some of you that may want to go on in genetics, I will help you by learning the mistakes that I made trying to do this in college. I never fully understood it until I started working with high school students on what was going wrong, what was I getting wrong. So I'll explain those errors to you in the next installment of this week's work. So that's it for today. There will be a short Edge Elastic quiz on Hardy-Weinberg. Just make sure you have it is open notes. Use your notes, and you should do well on it. That's it. Goodbye.